Our first speaker is Bhanu Sharma, and the stage is all yours, Bhanu.
Thank you very much. So, hello everyone and greetings. I'm from Aiza Trivandrum, India. And at the outset, I'd like to thank Evolution 2026 for the platform where we could discuss our science with colleagues from across the world. And so, my broad interest is to look into how organism lives in a heterogeneous habitat. So, like the habitat that organism lives changes across time, across season, and I'm broadly interested in understanding how organism survives, or what are the different mechanisms they use to cope up with this environmental variability. So the talk that I'm going to present today is the study that is done in a population of a butterfly. I work on insects, so population of butterfly that population of butterfly from this region called Kerala. Kerala is the southernmost part of India, and to begin, I'd like to briefly talk about the environmental uh, variability, the seasonality that we see in our study area. So this graph, I'll try to take the help of this graph to explain the the seasonal changes that we see in our area. So there's like distinct dry and wet season, immediate precipitation, and dry season is expected. It's highly desiccating, and wet season it's humid. And the changes between the season is not just with respect to the aridity, but the plants the larvae eats, the host plant that the larvae eats, and the visual background against which the butterflies or the insects develops. All those parameters of the environment changes across the season. Now, uh, so insects in general, they are vulnerable to desiccation. And my thesis is focused more on how butterflies cope up with the desiccation stress. So this study is also more focused towards the desiccation stress. And insects are vulnerable to desiccation. They are small, they are ectothermic, and like because of the high surface area to volume ratios, they are very vulnerable to desiccation. So this is a picture of a typical habitat that we see here in our area during dry and wet season. Dry season, you see, it's like brown background, and it's very dry. Wet season is very green, lush vegetation, a lot of green grasses, and so on. Now, my question, the first part of the talk asks how do butterflies cope with the seasonal fluctuations in the aridity? Now, when the environment is predictable, when the change in the environment is predictable, there are a lot of literatures that says that the organism could use cues and develop the phenotype that will be better in the forthcoming like coming seasons or coming time. For example, in case of butterflies, the larvae that is developing in larvae or the pupa that is developing in the dry season or the condition that reflects a dry season. Perhaps they might perhaps they might develop a physiology or the morphology that might help the adults, the emerging adults, to cope with the desiccation stress depending on how what environment they were developed on. So developmental plasticity could be a one way of coping up with the seasonal fluctuations in the aridity. So we wanted to test this. We hypothesized that the larvae that is developing under dry season condition perhaps develops physiology that helps them survive desiccation stress better than those develops under wet season condition, and we predicted so. And to do this experiment, what we did is our assay was very straightforward. We reared larvae in dry and wet season condition, and once adults emerge, we exposed the adults to desiccation stress by exposing the butterflies in such a container, tight sealed, and then we checked how long this butterfly survives in those desiccating conditions. Now, as predicted, what we saw is that the butterflies that was reared in dry season dry condition, they survived longer, irrespective of whether it was males or females. And from this, we could get an idea that perhaps the developmental condition that the larvae are experiencing, perhaps is playing an important role in deciding how good you do when the adults are exposed to disease intolerance. Now, when I first showed this plot here, then I didn't put this butterfly wing wings. But what we know is that this butterfly also shows this seasonal polyphenism in terms of the wing morphology. So during dry season, what happens? During dry season, the wings of the butterflies, they don't have any eye spots. Here I have kept a forewing, and this forewing doesn't have any eye spot. And during wet season, what we see is the butterflies with a prominent eye spots on the wings. Now, this high spots, it's not just presence or absence, rather it's a gradient. So we do see that, say, if you look into this, then we see that from not having from absent eye spot to a moderate, intermediate, and the big prominent eye spots in both forewing and hindwing. Now, so there are hypotheses and the experiments that tested the adaptive function of these eye spots in the butterflies, and this the hypothesis was supposed the hypothesis was uh, said to have these eye spots supposed to have anti-predatory functions. So that is, during dry season, when they don't have eye spots, it is expected that this butterfly wings will match the leaf litter dominated background, hence get camouflaged. During wet season condition, this eye spots will rather act as a deflection, rather act to deflect the predator's attacks towards the less vulnerable part of the body, towards the wing, rather than on the head of the butterflies. So the role of the seasonal polyphenism in predator avoidance has been hypothesized and tested in several such Tarvin butterflies. Now, I also wanted to study, because if we look into this, then what I saw is that the butterflies that are here in the dry season condition, they seems to develop higher desiccation tolerance. And what we also see is that during dry season condition, we see that the butterflies are very small or absent. They don't have eye spots at all. So what I did was, in addition to doing the desiccation assay, I also quantified the eye spots, and to do that, I took the pictures of the butterflies, and then I took the proxy of the wing size as shown here by a red angle, and then I measured the eye spots. And then I plotted it, and what it seems is that the butterflies that are here in the dry season condition seems to have a smaller eye spots than one that is reared in the wet season conditions. Now, we also wanted to explore if there is a correlation between expression of the eye spot and the desiccation survival. So what we predicted was that with increase in the eye spot size, the physiological tolerance to desiccation might be reduced. I mean, the relation between the eye spot expression and the physiological tolerance might be negative. So what we saw was that irrespective of whether it's a male or a female, the individuals with smaller eye spots or smaller eye spot, they seems to do better under desiccation stress than those having a bigger or relatively bigger eye spots. So and this we did the modeling with keeping a body size as a cofactor, and we could find that the eye spot is still explaining this result. And we also did structural equation models, and from this, what we could understand is that the relative humidity independently also affects. I mean, relative humidity here means the desiccating condition, dry condition, independently affect the survival, and uh, independently also impacted the survival, and by a body weight also it impacted the survival. Now, to answer to this first question that I started the first question that I started with, how do butterflies cope with the seasonal fluctuation in aridity? Perhaps developmental phenotypic plasticity, that is mediated by body weight, is the way. And the second part of the information that we get from this study was that perhaps in addition to a seasonal polyphenism aiding in predator avoidance, the dry season moths perhaps has higher desiccation tolerance, and this eye spot expression might have a dual function. So at this stage, we don't know exactly how what links eye spot size to desiccation tolerance. And there could be several mechanisms. It could be the expression of cuticle hydrocarbon that might differ with respect to the eye spot expression. It could be how hormones are regulating eye spot size and desiccation tolerance. Or there could be the metabolic trade-off that we have not that I didn't explore in this study, but this metabolite and cuticle hydrocarbon and the how hormone could be playing the role. That is something that we are trying to explore in this now. I mean, in the current experimental setups. So with this, I'd like to thank my lab mates, my professor, Vlasa, and Evolution again for the platform, ISO Trivandrum is my institute, CSR for the funding, and thank you very much.
Thank you, Bhanu.
Yeah.
And we are open for questions for Bhanu.
Yeah. Is that good?
I have a question.
Yeah, but unfortunately, I can't see. I think there is some issues with my thing, but yeah, please, yeah, please ask. I can hear though. Yeah.
Okay. You're still sharing your screen. I don't know if that's why you can't see.
Could
But
be. Yeah. Yeah. Can I stop sharing now? Oh yeah. I think now I can see. Yeah.
Sure.
Okay.
Okay.
Yeah, I can. Yeah.
So, Bhanu, thank you for the talk. I'm curious, what's the lifespan of these butterflies?
So larval stage. I mean, if I talk about, let's say, if I rear them in 27 degrees Celsius, and let's say 85% is relative humidity, then it goes to the larval stage goes to 25 days to 35 days. I mean, that's 35 days is the maximum.
Mm-hmm.
And pupal stage might go for 6 days in average,
Mm-hmm.
5 to 6 days. Yeah. And adults, I've reared them for more than 45 days in the lab
Okay.
setup. Yeah.
So the ones you find in the wild that have eye spots or no eye spots, are they different generations, or can the same individual change between?
Perhaps not. Yeah, perhaps not. It seems that it's irreversible. So once they determined the eye spot, it's not. It cannot be reversed. So yeah. So
Mhm.
yeah.
That's cool. So do you think, I mean, what's known, is there developmental plasticity then that responding to desiccation, or?
So in our lab itself, one of my colleague did study. What he did was he manipulated the relative humidity. He kept the temperature constant and manipulated the relative humidity to create the gradient of aridity. I
That's
mean
cool.
the desiccating condition, and he also manipulated the host plant. So during dry season, maybe it's not just how dry the environment is, how
Mm-hmm.
dry the plant itself that might matter
Mm-hmm.
a lot. Perhaps larvae might be using the physiology of the plant itself as a cue about the seasonal changes. So what he found was that both the aridity, in a sense, when he manipulated the humidity, that in addition, the plant quality. If he feeds larvae with dry plants and rear them in dry condition, they seems to have smaller eye spots or absent eye spot compared to one that is reared in low humidity but normal fresh plants. So it seems that there is a interacting effects of host plant quality and RH, relative humidity.
Thank you.
Yeah.
Thank you, Bhanu. Jack, you can start. Oh, Anthony, when we are taking your question, Jack, you can start sharing your screen. And Anthony, Mark, can
Yeah.
you unmute Anthony? Anthony, you're unmuted. You can.
Yes, sorry. Do you know which genes are different or differently expressed between these different phenotypes?
I do remember that there are studies, but at moment I'm not being able to recall. But I suppose there are studies that have looked into the genes that regulates the spot expression. But yeah, I'm not being able to recall that for now.
Okay, great. We are running out of time. So thank you, Bhanu, for your great talk. Our next speaker
Thank
is Jack. So Jack, the stage is all yours.
Great, thank you. Yeah, so let me know if there are any issues with audio. A bunch of guys with leaf blowers just appeared outside. Okay. Okay, so I'm going to talk about local adaptation in oceanic field crickets. And like in other field cricket species, the males of the species have these sound-producing structures on their wings that allow them to sing when they rub their wings back and forth. And so that's a very strongly sexually selected signal, which is very important in mate attraction. And so my title slide mentioned that I'll be talking about Hawaiian populations of this species. So we think those diverged from populations in the core species range of Australia about 800 or slightly over 800 years ago. And they did very well for a few centuries until much more recently. We think about 40 years ago, a parasitoid fly was introduced from the mainland of America. And this created an interesting interaction because the fly is a specialist parasitoid of field crickets, and it uses their song as a phonotactic cue. And this is exactly what it does in Hawaii. So the gravid female will hear the male singing and will deposit parasitic larvae on or around that singing male. And those parasitic larvae will burrow into and devour that male cricket, which will kill it. So there's obviously very strong selection on the cricket populations imposed by this recent introduction. And although neither species is native to Hawaii, and we're not aware of them co-occurring anywhere else in the world, there's this introduced coevolutionary interaction where the fly needs the cricket to reproduce, and the cricket needs to evade this fly in order to survive. So researchers studying these Hawaiian populations kind of asked the question of how they would respond to the introduction of this existential threat. And what they observed quite quickly was the emergence and rapid spread of these mutant wing phenotypes. In this case, the sound-producing structures on the male's wing are drastically reduced or even lost. And that means that when these males try to sing, they don't produce signal at an appreciable amplitude. So they're protected from being located by this parasitoid fly. And these are referred to as flat wing phenotypes. Within the last decade or so, there's also been the observation that several other mutant wing phenotypes are present. And for a variety of reasons, these other mutant wing phenotypes all reduce the amplitude of male song to the extent that they are protected from being located by this parasitoid fly. So there's been this rapid response through genetic adaptation and recurrent genetic adaptation. So I'm going to talk about flat wing and curly wing phenotypes today, which I think are two of the most common male silencing phenotypes that persist in these wild populations. So we find crickets reliably at these 11 locations across 3 islands. And this bar plot shows differences in the frequency of this selection of male wing phenotypes across these different populations. And there's too much information here to digest, but the crucial point is that these yellow bars represent the frequencies of this ancestral phenotype. So we think these are males that don't express any of these mutations, are able to sing at an ordinary level. So the yellow bars are essentially, we think, representative of the proportion of males that can sing in each of these locations. And you can see there's a lot of variation, which is in itself surprising because we know this parasitoid fly is present at each of these locations. So I'm going to talk about work we did in 2022. I'm going to start by talking about what might influence the spread or distribution of these different wing phenotypes, and move on to talking about some of the consequences of rapid adaptation in the system. So we started looking for evidence of local adaptation by first trying to quantify the selection against male song in each of these locations. So we played back cricket song and we recorded the number of flies that were attracted to that song. And we used this and some prior knowledge and rampant speculation to infer at each location whether we think silent phenotypes would be favored due to selection from the fly. So these are points above this dashed line. or if singing-capable phenotypes should be favored due to their advantages in the context of sexual selection. So those are points below the dashed line. And again, you can see there's a lot of variation, but there are, for example, two populations clearly where silent phenotypes we think should be favored due to the fact that we caught a lot of flies when we played cricket song. And then there's this population where clearly we think singing-capable phenotypes should be favored because we caught very few flies. So we caught, I think, 1 fly in 5 nights of surveying at this location. And we used these observations as input into a genome environment association test. So our predictor variable is the strength of selection against song at each of these locations. And our response is allelic variation across the cricket's genome. And we observed that there's a strong signal, significant association between genetic variation in this narrow region of chromosome 2 and the strength of selection from the fly. Which could be some indication of local adaptation. So we cross-reference this with what we know about the genetic architecture of these two common male silencing phenotypes. The flat wing localizes to the X chromosome, and there we don't see any signal of selection. Curly wing, by contrast, localizes to chromosome 2, which is an autosome, and the strongest associated genetic variant is within the region of this gene, abdominal A. And that's exactly where we see the peak of association between the strength of the selection and allelic variation. So we think this indicates that we're seeing evidence that selection influences the frequency of this curly wing phenotype across these 11 populations. So it's interesting that we see evidence of selection influencing the frequency of one adaptive phenotype, but not the other. There are lots of potential reasons for this, but one that I'm interested in is the possibility of selective interference. So we know, for example, if a male cricket is protected from the parasitoid fly because it expresses this curly wing mutation, that its genotype at a second locus, so say this flat wing mutation, is essentially irrelevant. Whether it expresses flat wing or not, it's going to be protected from the parasitoid fly. So selection on that second locus is sort of shielded, it's hidden. And you could reverse this, but the key point is that these mutations can be co-expressed, and so it's not always clear how selection will act on one but not the other. But we've investigated this through simulations. So we've introduced either one adaptive mutation or 2 adaptive mutations that confer exactly the same adaptive benefit. So they're completely redundant. And we find that if you just introduce the one mutation, it spreads quite quickly through the population, but it spreads much more slowly if there are 2 mutations due to this pattern of selective interference. So that's all I'm going to say on that first point, and I'm going to move on to talking about some of the consequences of selection and genetic adaptation in the system. So we started by inferring changes in effective population size using our genome sequencing data from these crickets. And what we see aligns quite closely with our expectations. So we see, following the divergence or the inferred timing of divergence from the Australian mainland populations, we see this strong decline in effective population size, which is consistent with expectations of island hopping and repeated founder effects. And then we see this gradual recovery until the approximate time at which Formiocracia, this parasitoid fly, was introduced, when we again infer that there was a strong decline in effective population size. And then, as you can see from these error intervals, the resolution in recent generations is not great. So we investigated this further using a linkage-based approach. And here we see essentially the same trend. So across these different populations, starting with the inferred timing of the introduction of the parasitoid fly, we see these consistent and very strong declines in effective population size. So these methods obviously rely on lots of assumptions, but I will say they correspond fairly well with our observations in the field. In particular, our 2 most recent visits to these locations in which we've not observed any crickets, which I think is the first time we've visited populations and just found crickets to be completely absent. So I think this supports strong recent declines in these populations. But what I haven't mentioned is that those inferences are based on genetic variation on just 3 chromosomes. The reason for that is that across the remainder of these 11 chromosomes, we see evidence of really big structural variants, which I think are inversion polymorphisms. So where I've drawn these red bars is where I think there's a very large inversion, and that's based on discrete patterns of elevated linkage, as well as this sort of symptomatic clustering of samples into 3 groups usually. So where the central group should be the heterokaryotype group. And it's surprising that all of this genetic diversity has been maintained in these polymorphisms, despite these drastic recent declines in population size and the recent response to selection. So we investigated this by comparing genetic variation in these Hawaiian populations in green with genetic variation in Australia, which we think, these 2 metapopulations diverged over 800 years ago. And we see that at least for some of these inversions, the ones that I've highlighted in orange, it looks like those inversions are also maintained as polymorphisms in Australia as well as Hawaii. So I think that supports the idea that these things are being maintained by very, very strong balancing selection in these regions. So to sum up, we see mixed evidence of local adaptation influencing these wing phenotype frequencies. We infer very strong recent population crashes, but we see that a huge portion of the genetic diversity that's retained is in these inversion polymorphisms. And I think that's really interesting because we don't know a lot about the consequences of genetic diversity in these regions for things like adaptive potential, resisting inbreeding, depression, and so on. So I would like to thank these people. Thank you very much for listening, and I'm happy to answer any questions.
Thank you, Jack. People can either raise their hand or we have time for 1 or 2 questions. Yeah, Anthony.
Okay, how were signatures of local adaptation studied? Which statistic was used? So we used that genome environment association test. So we used the strength of selection from the fly based on the number of flies that cricket song attracted at each location as kind of the environmental variable. And we used a latent factor mix model to look for an association between the strength of that selection at each location and allelic variation across the genome. So does that answer the question?
We have another question from Anthony. Do you want to unmute yourself, Anthony?
Yes, thank you for the talk. Since they cannot sing to attract females, how do these males mate? Do you observe new behaviors or new adaptation?
Yeah, I think we get this question every time we give a talk, and it's a really good question, and I don't have an answer. I think there are a couple of things. One is that there are effects of the sort of density of cricket song on various behavioral phenotypes, which I think is an ancestral kind of reaction norm. So if you are a cricket and you don't hear many other crickets, you assume you're in a low density population. So maybe you move around more, you're more likely to disperse. And so at least that first response, the fact that they seem to move around more, could increase rates of sort of encountering conspecifics. I think the other point I would make is that we don't know that they do. So it could be that there's this introduction of the parasitoid fly has created a kind of evolutionary trap where these male crickets have lost the ability to sing because of the really strong selection against song, but that has long-term negative consequences for the persistence of these populations because it does, I'm sure it reduces the reproductive output of these populations. And so on the one hand, we don't see evidence of like a strong evolutionary recovery after the spread of these adaptive phenotypes, but it could also be that these adaptive phenotypes are directly contributing to the declines that we're seeing because they're reducing the rate of encounter between conspecifics and potential mates.
Thank you, Jack. And people can ask questions in the Q&A, which you can find in the More button, and you can pin in your screen as well. Rebecca, if you want, you can start sharing your screen. And so our next speaker is Rebecca Tarvin. And Rebecca, you can start.
All right. So I'm going to present a paper that was recently published in Ecological and Evolutionary Physiology about the genetics of aestivation in amphibians. So amphibians are a diverse vertebrate group. There are now more than 9,000 known species. And they have, sorry, they have a global distribution, except in the driest parts of the world, including some deserts and the poles. Here I've outlined the most arid regions on Earth, and you can see even though for example, the Sahara, there are few amphibians, in lots of these regions, there are a bunch of amphibians that have adapted to this dry environment. So amphibians in these environments face really serious challenges. They have to stay moist, given that they use gas exchange across their moist skin. They have to survive these hot temperatures, and then they have to have successful reproduction in what is usually a very short active season when the climate is suitable for them to survive. So in order to deal with some of these challenges, many amphibians in arid regions can go dormant. Some species can be dormant for several years. They can drop their metabolic rates to 30 to 10% of normal. This is called hypometabolism. They usually go underground to escape the heat and the dryness. Some of them can produce cocoons, like you can see here in the picture of Cyclorena, and they can cover themselves in lipids that prevent water loss. They can also increase osmolarity of their body fluids to prevent evaporation. And as a consequence of their immobility and lessened amount of available energy, often many of their organs shrink, sometimes drastically in size. And then after all of this, they have to recover rapidly to take advantage of seasonal rainfall to reproduce immediately, and then their larvae have to develop rapidly to avoid desiccation in drying pools. So many amphibians do this amazing feat of hypometabolism in arid environments called aestivation. Aestivation is a type of dormancy, and actually we know a lot about dormancy in general, but most of the studies are about hibernation in mammals. So we know that some things are shared between things like hibernation in mammals with aestivation amphibians, like changes in intracellular signaling, reversible protein phosphorylation, and also a global reduction in transcription and translation to save energy, and specific upregulation of genes that aid in the aestivation phenotype. I'll note that a lot of the adaptations present in animals that have these hypometabolic traits are useful to study for humans because they can give us new ideas about how to deal with human disease. Probably the most obvious one is dealing with muscle atrophy in patients that are comatose. Lots of animals go motionless for months or even years, and they don't experience the same sort of atrophy. In amphibians specifically, we reviewed the amount of research on genetics of aestivation, and we found there are 39 studies. They've been focused on total of 16 species of amphibians out of about 1,000 known estivators, so not a great representation. Most of these studies focused on protein phosphorylation, glycolysis, and activity of specific transcription factors. I'll also note all these frogs that estivate are super cute. And so a lot of these studies were done in the 90s and were targeted on the activity of specific enzymes or genes. So in fact, only 2 of these studies used untargeted RNA-seq methods, and the rest of them sort of had a priori hypotheses. So our goal in this study was to take an untargeted approach to be able to identify additional candidate genes and processes that underlie this amazing adaptation. And then we aimed to look at whether these processes were highly conserved or derived, and whether we were able to identify some novel processes with this untargeted approach. So at the time of the study, there were 31 genomes available with NCBI annotations, and for which there were data on aestivation. So our data set included whole genomes for 11 estivating species and 20 non-estivating species. And even though it's not many species, actually the phylogenetic diversity of amphibians is captured pretty well in this data set. So we use a method called RER Converge. This is a really cool program executed in R that takes alignments of orthologous genes from all the species in your species tree, and as well as a phenotype tree. So this is a tree where you'd label your foreground branches. So in our case, any species that aestivate. And then what the program does is it builds gene trees for every single gene. And then it looks, if you look at the right here, this is a distribution of branch lengths. And so what it does is it asks if your foreground branches, in our case, the species that aestivate, if on average, their branch lengths are longer than the rest of the branches in the tree, then that gene has a statistical association with the evolution of that trait. So if the branches are relatively longer, that suggests that there's been a lot of changes in the coding sequence. If the branches are relatively shorter, then that suggests there's been relatively few changes. So it's sort of a gene-wide estimate of positive and purifying selection across the entire genome. And it runs really fast in R. So I definitely recommend this method. So I'll note that RER provides information on coding sequence evolution. And like I mentioned, most of the prior studies on aestivation in amphibians have focused on gene expression. So these are sort of complementary ways to look at genes involved in this trait. So once RER provided us with sets of genes that were decelerated or accelerated in our focal species, we then plug these sets of genes into string, which looks for interacting proteins within those gene sets. And then we also use GO-term analyses to look for enriched processes, functions, and cellular compartments. And then we did a literature review and we compared our results to what was previously known. So as I mentioned, we used 31 genomes. We fed RER Converge over 13,000 gene alignments. And RER Converge identified 165 genes with accelerated rates and 158 genes with decelerated rates in our aestivating species. So here on the left, I'm presenting the string results, and on the right, these are GO-term analyses results. So for genes that were identified with decelerated rates, consistently we found sets of genes related to oxidative phosphorylation and translation in these groups. For example, the most decelerated rate was this gene called RRM2, which is essential for mitochondrial respiratory function. And this is relevant for aestivation because estivators face sort of two major issues when they're hypometabolic. One is that they have consistently decreasing energy reserves. And so they have to be wise with how they use their energy to their lipids or sugars to produce ATP. And the other is that they have reduced levels of oxygen available in their environment if they're underground, and also in their tissues, given the low rates of blood flow. And so in vertebrates, a lot of vertebrates will shift towards oxidative phosphorylation, which produces a lot of ATP for a small input. But in amphibians, some species have been shown to switch to glycolysis given the low amounts of oxygen available. For accelerated rates, we consistently found processes related to macromolecule transport as well as development. So if you look here on the right, you can see above cellular processes a whole bunch of GO terms associated with different types of transport. This was one of the novel things that our study found. For example, this trap complex was the second highest accelerated rate. And it's involved in vesicle docking at the ER and Golgi apparatus. I really enjoyed learning about this. These sets of genes, which help, for example, endosomes merge with Golgi. So any sort of intracellular transport of proteins or macromolecules, TRAP is helping sort of tether the vesicle to the membrane prior to its fusion. There's other genes that we found as well, like SAP30, which recruits histones, deacetylase complexes, and represses chromatin structure. So this is probably one of the players in the global suppression of transcription and translation during aestivation. And this was found previously to be upregulated in aestivating frogs. One of the overwhelming signatures in our data set was that there was a positive selection on processes involved in intracellular trafficking. And this is novel. This is not fully appreciated prior to the study. So these are just a number of different genes and processes involved. At the bottom, you'll see here the trap complex and the snare complex, which work together for that membrane fusion. And all of the genes on this diagram here, except those with asterisks, were shown to have significantly accelerated rates. So if we compare to prior literature, again, like I said, most of the prior studies were looking at studies of gene expression or measurement of enzyme activity. And we can kind of think about this in if you have an accelerated RER, then that gene across all of your focal species, in our case, estivating species, there's a lot of changes in the coding sequence. And when that happens, usually it's indicative of a change in function. And so if that gene has a change in function in estivators and it's increased expression or activity during estivation, we might hypothesize that that gene has a novel function in the estivation state. In contrast, if its activity is decreased during estivation, then it would be increased in the active state, and that would suggest a novel function in the active state. So genes that fall in this category, in the first category, range in function from development to translation and phosphorylation. And genes in the second category are often involved in transcription regulation. So what that suggests is having efficient transcription is probably very important for frogs and other amphibians to break out of that dormant state and be able to reproduce and develop fast. So in contrast, if we have decelerated rates with increased activity during estivation, then again, this means that there's relatively few changes in the coding sequence and thus purifying selection. So this would suggest that these genes are involved in core processes necessary for estivation. And again, the flip side of the relative comparison is higher activity in the active state, then it's probably involved there. So here we found genes related to protein synthesis and modification as conserved core processes necessary for estivation, and core processes necessary for the active state included a lot of processes involved in energetics. So again, sort of reflecting this need to quickly recover from a dormant state to reproduce. So just to summarize, we found that some processes were highly conserved, some were highly derived, but actually, although I didn't really touch on them, most of the processes had mixed patterns, with some genes being highly conserved and some being diversified across the species. So probably what's happening here is there's a set of highly conserved core processes necessary for the estivation state, and then a diversification of other processes that are related to species-specific ecologies. And we were able to identify novel genes associated with this trait. And I'll point out that even though we were focused on thinking about dormancy, actually a lot of the results point to what's important for being active. And I'll finish by just saying that estivation is an incredible trait. We think it's ancestral to amphibians, and it might help them continue to survive changing climates. And with that, I'll thank you for your attention.
So our next speaker is Isabela Hernández Rodríguez. So Isabela, you can start.
So, hello everybody. I'm Isabela Hernández, as it was already told. I'm a third-year PhD student at Martha Muñoz Lab at Yale University, and today I'm very happy of being talking to you about my project of unpacking a Caribbean adaptive radiation, looking at the ecophysiology of Hispaniolan reef frogs. So, to start, I want to take you into this immersive activity of what is living like a frog, right? So imagine you're a very vocal organism that at the same time is jumping around for its locomotion and is pretty much dependent on water sources. But also, your activity is mostly based on the fact that you might be nocturnal and you are an ectotherm, which means that you don't produce enough metabolic heat to warm up yourself, and because of this, you need of environmental temperatures to do so. But you're a wet skin ectotherm as well, which means that you need to keep your skin moist at all times to have good gas and water exchange. But despite of this, you have managed to diversify it in all these different microhabitats, so ranging from arboreal, semi-arboreal, fossorial, and so on. And by this, let me introduce you to the Eleutherodactylus group. So this is a diverse radiation of around 240 species that are ranging from the northern part of South America to the southern part of North America. But the most striking diversity of these groups occurs in the Caribbean region, where you can find 85% of this diversity, which is amazing. But if we zoom in into the Caribbean region and we find ourselves in the most geographically complex place in the Caribbean, which is the Hispaniolan island, the one that is shared between Dominican Republic in the east side and Haiti in the west side, you will be finding yourself at 3,000 meters above sea level and 46 meters below sea level, which is quite amazing. But this is not only impacting the geography, but as doing so is impacting the climate as well. So this is a very climatic heterogeneous place. In all these high elevation places that I told you about, you can find lower temperatures that in comparison to the rest of the island that you can see in this map of the Dominican Republic is very warm all the time. So despite of this, Dominican Eleutherodactylus, they are ranging all elevational gradients. They are from coastal level to the highest peak 3,000 meters up above sea level, and also they are encompassing all the microhabitat specialization that I told you about. So this microhabitat specialization is striking and is shaping their phenotype, but also this is constrained maybe by the fact of all this climatic variability of this elevational gradient that I told you. So my questions are, how does microhabitat use shape the evolution of thermal and hydrophysiology in these frogs? And also, what are the macro and microclimatic conditions that influence this physiology? So starting right away with my first question on microhabitat uses shaping their physiology, I want to start telling you a little bit of my methods. Of course, I go do field work, I capture my frogs, I take a myriad of environmental and climatic measurements, and then I take my frogs to a lab setting using a field metabolic system, and I perform water loss rates experiments on them on a flow-through respirometry system, and also I measure the thermal tolerances that I'm gonna explain to you a little bit further on. So to start on the same ground, tropical frogs are thought to follow the principles of Jansen's hypothesis. And when I say Jansen's hypothesis, I mean, if we look at the y-axis in here, looking at the temperature and altitude in the x-axis in tropical environments, we can see that temperatures are at low and high altitude are not overlapping because of the seasonality of the tropics. And because of this, organisms tend to have narrow physiological tolerances. So what I mean by this is that if we take my elevational gradient, the frogs that live at coastal level maybe are going to have very different physiological tolerances than those that are living in these other spaces of the elevational gradient. So for example, if we take the thermal tolerances of these frogs, focusing on the cold end, which is the CT mean, so the coldest temperature that the organism can withstand without losing their locomotive capabilities, and the CT max, which is the hotter end, so the heat tolerance, the hotter temperature that the animal can withstand without losing their locomotive capabilities. What we're expecting to see is a pattern like this: high altitude, high CT mean, and also, high altitude, low CT max. But elevation is not the only factor. Habitat variation also shapes physiological responses as well. So in this case, let's look at water. Now this plot is showing the Cruz and Piedrahita very neat paper of 2017, and you can see that they have a habitat variation in the x-axis and evaporative water loss in the y-axis. And what they found is that if we focus on the ecological habits now showing you the arboreal habit right here, you can see how frogs that live in drier environments are losing less water than those that live in wetter environments. But then if we plot the terrestrial frogs, they are losing much water than the arboreal ones by large. So if you're finding yourself in a shaded, homogeneous, wetter environment versus a sunny, heterogeneous, drier environment, this is going to shape your physiology in different ways. So let's start with the results. First, the heat tolerance. Here, I'm showing you a continuous straight map. You can see how there's a wide variation in heat tolerances across the Dominican Eleutherodactylus frogs, ranging from 32 degrees to 37 degrees. Warmer colors are higher values, and cooler colors are lower values. But now, let's look at cold tolerance. This is even more variable than heat tolerances. You can see how we have frogs that withstand 2 degrees, and we have others that go as low as 12 only, which is pretty weak, right? So if we try to make sense of this, maybe we can cluster them by microhabitat specialization and see if we see a pattern. And by the colors of these points, you can see that there's some clustering, but it's not quite straightforward. So let's unpack this a little bit further. So now I'm going to show you a couple of plots looking at the elevation in the x-axis and the thermal limits in the y-axis. So starting with the CT mean, so the cold tolerance, and looking at the colors as the microhabitat specialization, what we're expecting to see under Jansen's hypothesis is a pattern like this. Higher elevation, colder ends, so they withstand colder temperatures. And this is exactly what we're looking at by large. All our microhabitat specialists seem to be following this pattern, but not everybody. We have some microhabitat specialists that are doing their own thing, and we're going to unpack this further. Then if we look at the critical thermal maximum in the y-axis, so the heat tolerance by elevation, once again, under Jansen's hypothesis, this is our expectation. So higher elevations, lower heat tolerances. And this is what we're seeing by large. All our microhabitat specialists seem to be following this pattern to some extent, but there are some that are kind of staying the same, which is in this case the semi-arboreal frog. So what we think is happening is that species that are living in buffer microhabitats display lower thermal tolerances. And by living in these buffer places, they are diminishing the thermal variability of their environments and also maintaining a high humidity in their substrate. But this is not the only mechanism. Something else that frogs can do, they can use their behavior to buffer thermal adaptation by selecting the substrate that gives them the optimum thermal and hydric conditions. Something else that I did was clustering this microhabitat specialization into two categories, so tree and ground. And what I found looking at the cold tolerance, ground being the blue and tree being the red, is that even by correcting by elevation, species that are up in the trees can withstand colder temperatures. Then let's look at water. We find a large variation in water loss rates. And this is cutaneous water loss rate, so it is accounting for the shape and the morphology of the frog. But despite of all this, we can see that we cannot actually cluster this pattern by any microhabitat or ecomorphological pattern. But what we saw is a strong pattern between physiological axes. So right here, I'm showing you the CT max, so heat tolerance in the x-axis and cutaneous water loss in the y-axis. And the colors, once again, microhabitat specialization or ecomorphology. What we see is that there's a straightforward pattern between animals that are losing more water are more heat resistant as well. So to summarize this, thermal tolerances are strongly linked to microhabitat use, but water loss rates are not. Frogs that are found in the trees can withstand colder temperatures. And then as heat tolerance increases, rain frogs tend to lose more water. Now, to unpack these cutaneous water loss results a little bit further, I wanted to explore my micro and macroclimatic conditions. So looking at my microclimatic conditions, now focusing in minimum humidity in the x-axis and minimum temperature in the y-axis, and now the colors are gonna be cutaneous water loss rates. What I did was clustering my sites into colder, warmer, drier, and wetter end. And we did not find a lot of frogs in the dry and hot end. And you can tell me you're cherry picking because there are other very empty quadrates in there. So I did this again with the mean humidity and mean temperature this time. And once again, drier, wetter, colder, and warmer. And there's nobody in the dry and hot end. So we wanted to understand what is going on in the drier and hot end. And we plot this together by looking at the VPD that tells us how dry the air is. So looking at cutaneous water loss in y-axis and the local maximum temperature in the x-axis, and now focusing in how dry or less dry the air is, what we can see is this beautiful pattern in which frogs that are living in drier environments are losing less water, and those that are living in just places that tend not to be that dry are losing a lot of water. Lastly, microclimatic variables are the ones that strongly predict cutaneous water loss, as well as heat tolerances, but microclimatic variables are the ones that explain cold tolerances the most, probably because we have lower temperatures on the ground at the micro level than we have at the macro level. And lastly, we perform a phylogenetic path analysis to understand the causality of all these measurements. And what we saw is that habitat tends to shape the climate that these frogs experience. And by doing so, the climate at the same time is shaping their physiology. So to conclude, rain frogs inhabit a wide temperature gradient as long as it's wet. Frogs found in thermally heterogeneous environments are more desiccation tolerant. The habitat shapes the climatic conditions, and that at the same time shapes their physiology. And lastly, physiological variation cannot be explained by a single factor. So rain frogs diversification can only be understood under a multidimensional perspective. And physiological evolution is shaped by multiple factors that operate across micro and macro scales. With this, thank you very much, and these are my acknowledgements.
Thank you, Isabela. We have time for one or two short questions, and by that time, if Raquel, if you want to share your screen, that would be great. Lauren, you can unmute yourself.
Hi, Isabella. That was a great talk. Thank you so much. I was wondering, what do you think are the biggest sort of outstanding next steps for you to bring this to a conclusion?
Yes, so I'm going to the field again this summer, which is kind of worrisome to me, but exciting at the same time. And something that I'm trying to explore is that I want to put sensors, long-term sensors, in each one of the microhabitats to see how temperature and water is actually behaving at the microecological scale. And something else that I did and I haven't analyzed yet is that I'm looking deeply into their ecomorphology. So I have CT scans that I perform already, and I'm gonna try to correlate their morphology into all this variability in the physiology that we're seeing. So I think we're up to something very exciting.
Sounds like it. Thank you.
Okay. The rest of the questions you can ask Isabela in the Q&A, which you can find in the more option. So our next speaker is Raquel Ponti, and you can start now, Raquel.
Okay, thank you very much. Well, I'm Raquel Ponti, and now I'm a researcher in the University Rey Juan Carlos of Madrid. But this research, I did this research during my Marie Curie at CBO in Portugal. So here we want to see how resource exploitation traits evolve on island endemic birds. So, sorry. Well, islands have particular features like low species diversity, also have milder climates and limited resources, and usually are small and isolated, as islands are. So all these characteristics make that there could be common patterns of adaptation of island species. That all these patterns can be englobed in the island syndrome. So there are, for example, morphological adaptations like body size evolution on islands where species can be bigger or smaller comparing with the mainland relatives, or species also usually are less aggressive on islands. And there are other kind of adaptation in life history traits or signaling, for example, differences in color, especially in birds. So here we want to see how island birds adapt on islands, but considering the resources that are available. So we expect that on islands also there's a reduced competition because since there are less species on islands, we also expect to have less competition between similar species on islands. So because of that, we expect the species expand their niches and became more generalist in terms of diet, but also maybe in foraging behavior. So different foraging types that they can exploit. And this will involve an increase in morphological adaptation of island environments, island species. So here we want to see how these resource exploitation traits evolve on islands. And in this case, we were exploring both beak shape and size and the tarsus length, because we think that those traits are the ones that are related with the resource exploitation. So to do that, we use 796 pairs of island mainland bird species. So this means that we use endemic bird species from all these islands that you can see here in the picture, and the closest relative in the mainland. So we can compare how evolved all these their morphological features from the mainland to the island. And we use all 1,170 species worldwide, belonging to 16 orders and 78 families. So in this case, to test how they evolved, we used these Bayesian phylogenetic mixed models, in which as dependent variables, we use the beak length and the beak depth width. In this case, we used both, we measured both measurements, but depth and the width were super correlated. So we did the models only with the beak depth and the tarsus length. In this, all these measurements were for part of the species were obtained in the museums. We measured them, and the rest for the species that we couldn't find the measurements, we used the Abonet database, that it's a database with a lot of measurements of species. So we first build some simple models to see how these variables, these measurements vary from the island to mainland. So just using the insularity variable. And then we use the latitude and body mass as covariate to control for them, because we expect that the appendixes could be different depending on the latitude. So in colder habitats, probably the appendixes are shorter because of the Colin rule, I think. And also we use the body mass as body size control to control by all the variability that is due to the body size, right? And we use the random effects of the phylogeny, pairs, and species. And then we construct other complex models in which we include all these variables, but also we included the competition pressure as the number of species of the same family, because we expect that similar species belonging to similar, well, the same family will have similar ecological requirements. And we use this Gini index, that is an index that measures the specialism, how specialist is a species. So here I'm gonna explain a little bit more about it. We use this Gini index for both first for diet breadth and also for foraging habitat breadth. So this coefficient goes from 0 to 1, where the 0 means that species are more generalist, both in diet and foraging, and values near to 1 means that are specialist. So we calculated these indexes using the frequencies of diet from Abonice, that is another database where there's, the diet, the frequencies of the diet per species are available for all the species of birds, both island and mainland, and we use also 32 levels for foraging habitats. So what we find is that here we have the complex model for the beak length. So we find first that the island birds have longer beaks than the mainland ones, you can see here. And also that more generalist species have longer beaks as expected. So here we have in the table the estimate for insularity for the simple models without all these variables here and with the complex one. So we can see that part of this, part of the variability is explained by this Gini coefficient for diet, because the insularity was reduced here, but there's a lot of variability in any case that is not explained by the Gini coefficient. So there are other forces on islands that are explaining an increase of the length of the beak that we will explore in the future. We found similar patterns for the beak shape. So for the beak, the depth, in this case, island birds have thicker beaks again, and more generalist species also have thicker beaks. But again, we didn't find differences in the estimate from the simple and the complex models. And finally, here we have the tarsus length model, in which again island birds have longer tarsi, and again the Gini coefficient for diet was related with the tarsus length. We expected also here to find some kind of indication of the foraging specialism to tarsus length, but we didn't find anything. It's not significant. So probably this is because of the type of diet instead of the foraging type that are increasing the tarsus length. So to sum up, basically what we find here is that longer beaks, thicker bills, and longer tarsi were associated with insularity and broader niches. So we can support the prediction from an ecological release theory. But for example, we didn't find anything regarding competition. So I didn't tell it before, but competition was not significant in any of the models. So this resource diversification on islands probably is because of the diet flexibility and resource limitation rather than reduced competition pressure alone. And also there's a lot of variability that is not explained by this, that if the species are a generalist or specialist on island. So probably there are other forces behind in which there's an increase of beak and tarsus on island, maybe not just because they become more generalist only, but also because maybe some of the species change their diet or the diet niche. So maybe there are a suite of niche diet of island birds, but also of the generalist type. So those are the conclusions, and if you want to know more about us, we have different project ongoings, also regarding comparative work on island birds, but also ongoing project in Sao Tome and Príncipe, Canary Island, and Azores. And yeah, if you want to know a bit more about our project, you can check our webpage, and we are open to collaborations and everything. So thank you.
Thank you, Raquel.